I'm probably going to. I do want to tell you a couple of quick stories about the extraordinary toilet habits of sap-sucking insects again, because I feel like that was promised. And this headline is one of my favourites that I've seen describing the common meadow spittlebug, Philaenus fumaris. This isn't Philaenus fumaris actually. This is the alder spittlebug, I think, Afrophora olmi. But they are, as I mentioned earlier, these sap-sucking insects have these choice of these two different plant saps, the phloem, rich in sugary and nutritious, and the xylem, which is watery and a little bit salty, and really there's not very much nutrition in there. And yet many of these insects choose to feed from the xylem, which is a very strange choice. Also, as I mentioned, the phloem is under positive pressure, and when you stick your stylet in there, it just comes out. The xylem is under negative pressure. That's because it's drawn up through the plant, through the force of evaporation from the leaf. So it's actually sucked up out of the ground. And if you stick a straw on that, it sucks against you with a force of 1 megapascal, which is a really strong force. So not only have they have to extract this really nutrient force out, they have to work really hard for it. And if you look at a lot of these xylem feeding bugs, they all have quite a large protrusion in the front of their head, and that's all muscle. So inside the head, they basically used sort of MRI scanner, but a little one on a bug. And they've looked at the internal structure, and inside this is all muscle, and it pulls on a membrane which enables them to extract that really nutrient poor sap. And when they do that, of course, as with the aphid, it has to ingest far more sugar than it needs, and it has to excrete all of these sugar. These bugs have to ingest such a huge volume of that sap to get sufficient nutrition that they have to excrete an enormous volume of liquid, equivalent to, I think there's a video I'll show you actually of a sharpshooter, 18 tons of water if they were the size of a human a day. Right, so that's a lot of excretion just to get the nutrition. What a weird lifestyle choice. But these, and you probably know these spittlebugs from the foam they make in the spring, which we know as cuckoo spit. It's one of the signs of spring, and that is actually produced from this vast volume of excretion they make. They put glycoproteins which act as a detergent, and then blow fart bubbles into it to make foam that they live inside. They're also fascinating because the spittle-bugs are actually pound for pound the highest jumpers in the animal world. They better than fleas, better than kangaroos. Because they're a couple of millimeters big, but they can jump 70 centimeters, something like that, which is probably equivalent to a human jumping 200 meters in the air, over a tower block, which is pretty incredible. They're not very controlled jumping. It's a great escape mechanism. They can also fly, but they're not very good at it. I've got a whole science story here about why we're interested in these bugs, which I'm going to skim over because science, but they have a dark side, that there's a disease they carry. This is another bacterial disease that infects plants, and it's causing huge problems in Italy because it's devastating the olive trees in Apulia in the south of Italy. And that's been a problem over the last decade or so. It's remained contained in Apulia and hasn't really spread much further. The bugs are they, like the mosquitoes, they carry the disease. The bugs are very common in the UK, but the disease is not. And obviously we don't grow many olive trees, but this particular pathogen, it has the potential to infect other plants. And this is really why we did a very large, hello.
What is the one that's common?
That does what, sorry? Yes, it's Philaenus spumarius, which is the common meadow spittlebug. It's very common, yes. It's all over Europe, but it causes no problems in the UK. It's a native, and it feeds on plants, but it doesn't do any damage, and there are no significant diseases in the UK that it carries. But there's a warning that people really need to be cautious about importing plants because that's the way that diseases move around the world. And we did a lot of public engagement around this and trying to make sure that people working in the plant trade industry and the nurseries knew about plant health registration. We did lots of science here. We understood gene flow. We made these maps. From looking at the genetics of these insects, we were able to trace their patterns of historic movement by looking at how the genes had moved around between populations. And we could see historically there are areas where the bugs have been very restricted in their movement and other areas where they've been able to move around freely. And we got these bugs from all over Europe, and we could see that, weirdly, we don't know what the basis, these brown areas are areas where the bugs don't move around very much. And the blue areas are where they seem to move much more freely. And we don't know what the reason for those particular areas is. But this is, DEFRA wanted to know this because if a disease arrives in the UK, and it hasn't yet, this will inform quarantine strategy. Because if it arrives in, somewhere in this corridor from the Wash, then it might spread quite quickly. But if it arrives in Devon, then it's going to move quite slowly. But I'm going to skim over that. We did have one guy. We had lots of public engagement of this. The spittle bug watch. We got people reporting sightings of spittle. The messaging got a bit confused because lots of people were phoning us up saying, "I've got cuckoo spit on my plant. Does that mean I've got the disease?" And of course, no, it doesn't. Cuckoo spit is a normal part of this insect's life cycle. The insect isn't a problem, and it doesn't mean you've got the disease. And one chap phoned up and said, should he be shooting cuckoos? So we had to tighten up our messaging a little bit about that. But as a result of our work, we got all of this advice coming out of DEFRA on what plants you should look out for and be careful importing. These are plants that might carry these diseases if they're imported. DEFRA and the Animal Plant Health Agency restrict the movement and they quarantine plants when they come in. I'm going to risk trying to play this video. This is another group of xylem feeding insects. These are leafhoppers, and the particular group of leafhoppers called sharpshooters. And they're called sharpshooters, surprise, surprise, because of their extraordinary toilet habits. And I'm going to see if this will play. Probably not. They excrete, they feed from xylem, they excrete a huge volume, and they don't want to get it on them. They want to get rid of it. So they've got this structure, which is like a flap that catapults droplets of their excretion out at enormous speed and over some distance. And that's why they're called sharpshooters. But unfortunately, we're not going to get the video of that. There's the rhododendron leafhopper, very beautiful, red and green. That's an invasive one, which followed. But they are sharpshooters. I've never seen them doing the thing. But one more extraordinary toilet habit, lerps. I've only learned about these quite recently. These are structures that are produced by psyllids. These are very close relatives of aphids. They feed on the phloem sap, full of sugars, and like the aphids, they have this problem of having to get rid of all of this sugar. But instead of making this liquid honeydew, they crystallize the sugar and they make houses out of it, out of their poo, their sweet, sugary poo. So they're these little like crystalline structures that they live inside. And they're, I don't think there are any of these in the UK, but they're really predominant on eucalyptus in Australia. And quite often you find showers of these things falling from eucalyptus trees on Australia, and they cover people's cars, and then they get crows coming to eat them, and they sustain ecosystems as well. The kids love picking them up and eating them because they're sweet. And I only learned about this week. There's the bell miner bird, which feeds on lerps, and rather like the ants and the aphids, it's got this relationship where it protects the psyllids from other predators, only eats their excretions. But this actually causes problems for the ecosystems, and we get this phenomenon called eucalyptus dieback because the eucalyptus are so heavily infested with these psyllids, because the bell miner bird are protecting them, that you get the trees die and the ecosystems die. So these birds end up being a problem because of their predelication for this weird insect. Anyway, this is where I'm going to the second part of the talk now, which is a different story, and it's a bit late for Halloween, but this is a story about zombies, zombie plants in particular, and I've called it Pint of the Living Dead because it also involves beer for a reason that you'll come to. And actually, part of the origin of this story went back to this post on social media. This back in 2018. And this chap posted a picture of his hop plants and said, can any plant types tell me what's going on here in terms of genetic control of flowering? Hop cone and others have leaves between the petals. And here you see, this is what a hop flower normally looks like. And this one's got leaves growing out of the middle of it. And Ben here was curious about this and thought this might be a mutation. And he tagged some plant scientists who might know about that sort of thing. And I saw it and I thought, I think I know what that is. And Tracy is Ben, because she works with him at Wymondham College, where he's head of biology. And I got in touch with him and said, I think I know what that is, and I'll tell you that. And this is we know this because I've introduced you to the group we work in, and that we work on this bacterial transmitted bacterial disease called phytoplasma. And that's what I thought this was, because phytoplasmas do very weird things to the plants they infect. And one of those things is they change the way that the flowers develop. And what you see on phytoplasma infected plants, for example, the Madagascan periwinkle here, the phytoplasma infected plants, all of the petals develop as leaf-like structures, and they're green and they're bushy. And you see similar things in all sorts of other plants. In the chickpea, the infected flower is green and leafy. In oilseed rape, in our favourite weed, Arabidopsis, the infected flowers just look like little bunches of leaves. And I thought Ben's hop plants might be an example of this infection causing the floral organs to fail to develop correctly. And what's going on here? Why do they do that? What are phytoplasma? Phytoplasmas are a group of bacteria belonging to this particular part in the family tree, the Firmicutes. They're obligate parasites. They can't, like the aphid symbionts, they can't live outside their host. They can only exist inside the plant cell. They're actually intercellular, but also they're transmitted by insects. So the sap-sucking leafhoppers will pick up the phytoplasma from the infected plant sap and then transmit it to other plants. So these bacteria have got the ability to live inside the insect or in the plant, completely different hosts. They can't live outside them. And they do not just change the floral development, they do other weird things to the plant as well. One of the things they do is they cause the plants to become incredibly bushy. And you can see that in a few examples. This phytoplasma infection in a pine tree produces, these are called witch's brooms, these types of symptoms, because they're just these really bushy, thick reaches. It happens in lime trees. This is quite a pest of lime trees in the Middle East, and this phenotype is they produce lots of leaves, they don't produce flowers, they don't produce fruit. It's a problem. And also, you may have seen it in poinsettia. Nothing to do with the red bracts and the beautiful colouring, but the bushiness of these plants is down to an infection with a phytoplasma. And the breeders who were making these ornamental plants didn't know that for a long time, and they were trying to breed these beautiful bushy plants, and then suddenly they'd find it disappeared and all of their plants were really tall and leggy and not very attractive until they figured it's an infection and they have to maintain it by grafting the plant so that the bacteria is transmitted and you get those. So when you've got a poinsettia in your house at Christmas, you've got a phytoplasma infection. So this is some more examples of the floral phenotype. These infected echinacea cone flowers, all of the floral organs have turned into these green structures. You also get something called indeterminacy. When you have a flower, it's what's called a determinate structure. It's always going to have the same form. You've got sepals, petals, stamen, carpels, thank you. And that's the plan, and that's what's going to develop. But these break this plan and think, no, we're going to keep producing new structures, and you get flowers growing out of the middle of other flowers, and they just sort of become more fractal-like. And we see that again in our little weed. We see just instead of a single flower there, you just get a whole bunch of them. And why are they doing that? This is the life cycle I mentioned. So we've got the phytoplasma infected plants with the symptoms here. If you look closely, this picture, there's actually a leaf hopper sitting there on that petal. And that's because they're not pollinators. They don't care about flowers. They're not attracted by the signals that attract bees. These are sap feeders. They like nice green bushy plants. And so actually these symptoms that the bacteria causes in the plant make them more attractive to the insects that are going to come and pick up the bacteria and spread the infection, as this meme shows. So yes, there you have the leaf box saying, no, I don't want the straggly, leggy, unbranched plant with flowers. I want this green. And I'm going to go and lay my eggs on that. And in our lab, we started thinking about this as well. The bacteria has taken over control of the development of the plant, and rendered it sterile so it can't reproduce. They actually live longer, these plants as well, especially the annuals. They die much slower, so the plant lives longer, so it's there to enable the insect to come and pick up. And all of these are features that we associate with zombies, right? So we call these zombie plants in the lab. Because they live longer, they can't reproduce, they only exist to spread their infection to other plants. And this is, there are plenty of other examples of zombie-like parasite infections in animals, mostly. I don't think I've got the animated version of this. Yes, I have. This is a fluke worm that infects snails, and it causes this weird flashing on its these are eye stalks, aren't they? Which just stand out a mile away, and this attracts birds to come and eat them. And the bird, of course, is the secondary host of the parasite. So just like the phytoplasma is making its host more attractive to its vector, the same is happening with this fluke worm. And this is not unique. We also have the cordyceps fungus, which is a fungus that infects many different insects. But perhaps most famously ants. And if you don't know about cordyceps, well, they're amazing. They infect the live ant. They take control of the ant behavior, and the infected ant will then, instead of doing its normal ant business with its colony, will just climb up to the top of a tall tree and then clasp onto the tree until the fungus has killed it. The fungus then produces a fruiting body from the ant's head, which disperses the spores, and because it's gone up the top of the tree, it enables much more effective dispersal of those spores. And of course, if you're familiar with the computer game and the TV series The Last of Us, this is all about a human zombie apocalypse, which is brought about by cordyceps fungus. This is another fluke worm which infects frogs and causes developmental abnormalities. Their legs don't develop properly. They're not very mobile. They can't escape predators, which I think are birds and badgers, apparently. And again, these are the secondary hosts of the parasites. So the parasite is again making itself more successful. And this one is it behaves a bit like the phytoplasma in reverse, because this is fungal infection, a Puccinia fungus, yes. And it infects, I think these are a brassicaceous plant. And these are not flowering plants, these are vegetative growth, but the fungus causes a mimic of flowers which attract pollinators, which pick up the spores of the fungus and spread them. So zombies exist, folks, and phytoplasmas are the cause of one of those. And the science bit is what we do in the lab is we've been working to understand how these phytoplasmas cause these changes to plants. So feel free to turn off now, because I'm going to get into some molecular biology and biochemistry, gene expression regulation and all of that stuff. But basically, when they infect the plant, they're inside the plant cell, and the phytoplasma bacteria secretes its own proteins out into the cytoplasm of the plant, where they get to work reprogramming the plant cell. We've identified getting on for 60 different proteins that these phytoplasma secrete into the plant cell. We've been doing experiments to find out what they're doing by taking the DNA from the bacteria that encodes those proteins and putting them into plants. And then, in the absence of the bacteria, we've been able to recapitulate the symptoms that the phytoplasma cause. So we could say, oh, we know that this gene from the phytoplasma causes this symptom. So the protein it makes is one of these. And then we've got to work finding out how they work. And essentially, we've identified, we call them SAPs, secreted asper-yellow phytoplasma proteins. And these two, SAP11 and SAP5, they both cause these plants to become very, very bushy. SAP54 is a really exciting one because we put that in a plant, just that one gene, no phytoplasma, it causes the plant flowers to completely change and become these leafy structures. And all of these, in fact, when we put these genes into plants with no phytoplasma there, it makes those plants more attractive to the insects that transmit the pathogen. So whatever they're doing, it's the changing look, the bushiness, the leafy flowers, the insects are like, yes, please, I'll have some of that. And how SAP54 works, I think, is really cool. Here we go, sepals, petals, stamen, carpels. This is the model. This is a classic piece of plant genetics from the 1990s, was understanding how flower development is controlled. And this came from a few groups, but including scientists at the John Innes Centre, who put together what we call the ABC model, where we have 3 genes called A, B, and C, which govern different areas of the petals and different combinations of these genes. If you've just got gene A, you're going to get sepals. If you've got gene A and gene B, you're going to get petals. And you've got B and C, you get stamens, and you've just got C, you get carpels. So these genes are expressed in different parts of the flower, and that governs what those organs turn into. And we can see if we mess with that, we're going to change what those organs are. And that's exactly what SAP54 does. And you can see if we make mutants in some of these genes, so we break gene B, we only have A and C, we're only going to get sepals and carpels, and that's what we get in this mutant. No phytoplasma here, no phytoplasma genes, just broken plant genes. But they start to do things that look like the effects of phytoplasmas. And this C function mutant, no C, so no carpels or stamens, loads of petals. They're very beautiful. And so what SAP54 does is it targets the proteins that are encoded by these plant genes. They're called transcription factors, and they regulate gene expression. And SAP54 gets into the plant cell and it starts degrading these transcription factors, and it causes this change in plant development. And I said to someone earlier, no graphs, so I'm afraid there's a graph here. This is showing how much more the insects like the plant with SAP54 in it. So when I saw this message from Ben, that's what went through my head. I thought, I think these are floral organs. These are not leaves. These are actually bracts. I think that I don't think these are petals in a pop organ, but I think there's a phytoplasma in there. And I think it's got a SAP54 and it's changing the development of these. So I replied to Ben, say, shall we have a look? And so he was coming to the John Innes Centre for a meeting, and he bought a big bag of hops and handed them over, and we did some DNA extraction and some PCR, which is amplifying a little bit of DNA. And we were looking for phytoplasma DNA. And from the symptomatic flower, we extracted the DNA, and we got this band, this bit of DNA, which says, oh, yeah, we can detect something here. But when we looked in a flower without symptoms, we didn't detect anything, and we purified that bit of DNA, and we read the sequence, the A's, the T's, the C's, and the G's of it, and then we searched that against a database, and it's a phytoplasma. So we were right, this is nice. So we thought, this is exciting. What are we going to do with it? Well, first we looked at the flowers as well as the petals. We thought about this indeterminacy. Do the flowers develop according to this normal pattern, or do they get out of control? And actually, it looks like they just keep producing more organs. The normal hop flowers are much shorter and smaller. I thought this is exciting because we might be increasing our yield, because of course this is the bit of the crop that you want. And of course we thought, well, we better make some beer with it, because it turns out Ben likes home brewing and I like home brewing, and my friend Larry also likes home brewing. So here's Ben and Larry in my shed with our homebrew kit, the software that Larry wrote that designs recipes, in a big bag of phytoplasma infected hops, and a bowl of malt there. And in fact, the malt, we thought, well, what should we do with this? And actually, this was about the time that my colleague, Chris Rideout at the John Innes Centre, he'd been investigating heritage barley varieties. So he was looking at varieties of barley that hadn't been grown since the Victorian time. But we've got a fantastic collection at the John Innes Centre in our germplasm resource unit, which includes all of these varieties. And he was looking for novel sources of disease resistance. And he found some in a variety called Chevalier, which it turns out has really nice malting properties as well. And he set up a company and started selling this variety. And we thought, well, this is being brought back from the dead, like a zombie. So it seemed like the obvious choice. And we made a stout and it was delicious. And that was the pint of the living dead. And I'm going to stop there. So thank you all for your attention.
Well,
And if you have any burning questions, then I'm sure.
Like we said, if you've got any questions. Did you actually produce any beer from the yeast of Petso?
Sorry. You know about that.
Yes.
That's a completely non-science driven project. But yes, I was talking to a friend about traditional brewing practices, and before people knew what yeast was, they quite often throw a bit of bark from a tree into the wort to kick off the fermentation. And we thought, oh, well, can we isolate yeast? You know, because we've got a lab. And we did, and I went for Kett's Oak because, well, what did the Kett brothers do there 500 years ago? They fomented a rebellion. So I thought, yeah, I want to ferment some, but it turns out next door to the John Innes Centre, where we've got the Quadram Institute, they have the National Yeast Collection there. And so I got in touch and said, do you want some yeast? And they said, yes, please. And they do a good job. They'll isolate it to species level for me. And they're keen for more submissions. And I just thought, wouldn't it be nice to collect lots of yeast from famous and ancient oak trees? So wherever I go, I've got a little yeast collection kit.
It's really annoying. It's not annoying, it's just like, all right.
But I've got about 30 different isolates from famous and ancient oak trees all over the country now. And I am going to do a brew. So I'm going to take the same base malt beer and do small batch ferments with 30 different yeasts, some of which I think are completely inappropriate for brewing. I sort of Google the species name and it's called the brewer's nightmare. But some of them I think will make something quite interesting, but in due course. So yes, I did get some yeast. I also got some yeast from the tree outside the conference centre at the John Innes Centre.
I think you've got something with the college as well.
Oh yeah, yes. Well, Tracy, you wouldn't let Ben send the petri dish by post.
No.
Did it not work?
No, unfortunately it died out, so we need to go back to that one and get it again. Yeah, yes. Yes, thank you for bringing that up. Okay.
I think it's a really hard question to answer.
No.
It's wonderful to hear people laughing about insects and getting them to use about insects.
Oh, good.
Yeah, just great.
Well, yeah, I hope I've persuaded you all that evening and got something to offer. Well, thank you all very much. It's been a pleasure.